Before any psychedelic experience, a billion of molecules of LSD need to travel your body. But how does LSD actually find your brain? And more importantly, how does it get out of there again? If these are questions that are interesting for you, stay tuned. Pharmacokinetics. So I said pharmacology has two columns. We got the pharmacokinetics and the pharmacodynamics. And the pharmacokinetic asks the question, what does the body do to the drug? So once it is intaken, what does the body do it? Where does it bring it to? What does it do to the molecule? How does it get rid of it? So it's basically about the journey of the drug through the body. This is what pharmacokinetics means. Kinetics means movement, and pharmacon is the drug. In pharmacokinetics, there are four main processes, which are often abbreviated as ADME. A stands for absorption. This process deals with the question, if I consume a drug orally, or I put it under the tongue, or I put it transdermally or rectally, how does it actually find its way into the bloodstream? Because only from the bloodstream it is actually able to end up in the brain or wherever you want to have the drug. So this is considered absorption. How does the drug find its way into the bloodstream? Once it is in the bloodstream, we deal with distribution, and pharmacokinetics knows different models of how to describe them. The easiest one is the two-compartment model, which assumes that there is a compartment where a lot of blood is, so the vessels themselves, but also highly perfused tissues. And there's another compartment, which is the peripheral organs, which are less perfused by blood, such as muscles, fat, and skin. And distribution asks, once the drug is basically in the blood, how does it find its way to the organs? Once it is distributed, but also already on the way to its distribution, a drug also comes across the liver. Some enzymes can also be found in the gastrointestinal tract and all kinds of other organs as well. And within the liver, the body tries basically to get rid of the drug. It would cut off pieces from the drug or add certain groups to the drug, so that it becomes easier for the body to get rid of the drug. And the idea is also to inactivate the drug by working enzymatically on it. So this is what metabolism means. And the last step is excretion. This is the question, once the body has traveled, once the drug has traveled our body again and again and again in hundreds of circles, how does the drug actually finds its way out of the body? And here, of course, kidney and the feces are the most important ways. And we will now go through these and discuss them with regard to LSD. This is the psychedelic I chose because we have most literature about LSD. When dealing with the pharmacokinetics of a drug such as LSD, we don't always have all the information for a certain drug, because understanding how one drug, it's a very small molecule, actually finds its way through the body is not so easy to understand. What can help, however, is if we know a little bit about the properties of the molecule. We know that LSD is a relatively small molecule. Here you see the molecular weight, which means it's more easy for LSD to travel the body because it can basically slip through the cracks, so to speak. We can also look at the logP value, which is around 3, which is a very good value. It basically means if you ask LSD, left is a room full of fat and right is a room full of water, where do you want to go, LSD? LSD would rather prefer to go into the fat room. Some molecules would also find their ways into water, but LSD is more lipophilic. We also know that LSD is actually a base. It's not an acid, although we sometimes refer to LSD as acid. It's a base. It's a weak base. And this means when it is in an environment where there's a lot of acid, such as in the stomach, it's actually protonated. And these values already can tell us something about the journey of LSD through the body. But let's go through it step by step, how this actually works. When we assume that we consume LSD orally, for example, by swallowing a blotter, then we can assume that LSD would travel down the esophagus and would eventually end up in the stomach. Some people might assume that LSD now is being absorbed from the stomach, but this is rather not the case because of its pKa value within the stomach. LSD molecules would be protonated, which means they accept an H plus proton, and this makes LSD charged. And when LSD is charged, it actually cannot travel out of the stomach because it cannot pass the membranes. That's why LSD is most likely transported into the small intestines. Here the pH is much lower, and here LSD molecules are less protonated. So here it's actually much easier for LSD to go through the endothelial cells of the gastrointestinal tract. It's most likely, given its high lipophilicity, that it travels through the membranes to actually get to the other side, but we cannot rule out that it also slips through the cells, through the endothelial cells, because of its relatively small size. Okay, once it actually left the intestines, it goes via the portal vein into the liver, which can be seen up here. And then it's basically carried along with the blood to the heart. The blood is then carried further to the lungs because the blood is meant to be added with oxygen. Then the drug along with the blood will go back to the heart. And here it's being pumped, so to the heart, to the lungs, and back to the heart. And from here, it is basically pumped, so from the liver to the heart, to the lungs, back to the heart, and then it's actually pumped into the system. And this pumping into the system works via the aorta, which can be found up here. And the aorta actually has multiple exits. So some of these exits would travel down the body, some of them would go back to the body, and some of them would actually go back, would go all the way up to the brain. And before the aorta actually enters the brain, it would actually emanate into the carotid artery. This then branches in front of the brain, and part of it would go to the face and the other part would go into the brain. And actually, after the drug has been absorbed, which is down here, as I mentioned before, we already speak about distribution. So as soon as the drug is being pumped into the system, we speak about distribution. This is what I told you before. The drug would be carried via the internal carotid artery to the brain. And here, the carotid artery would go multiple branches, such as the anterior cerebral artery, which carries the LSD to the frontal cortex. And this is one of the main regions where we believe that LSD is active. Now, we need to have in mind that LSD is transported via the blood. So for now, even if it's up here in the anterior cerebral artery, it is still within the blood. And the drug still needs to enter the interstitium. This is where the neurons are. So it actually needs to leave the blood to find its way into the synaptic cleft. And this is not so easy, actually, because there is something that is called the blood-brain barrier. And here you actually see how the blood-brain barrier works. Here you see a brain in a sagittal section. If we enlarge this little area, we can see it like that. This region, which I'm highlighting now, is basically the vessel. And here the blood flows, you see all kinds of blood cells. And within this blood flow, you would also find billions of LSD molecules. And these actually now need to go across these endothelial cells. And usually it's not so easy for a drug actually to cross the blood-brain barrier, which is composed of astrocytes. These are these ones here. And they are also composed of these proteins, which create very tight junctions between the endothelial cells. These proteins, which tie them very closely together, are called claudin and occludin, which already tell you that they are meant to really make sure that the endothelial cells are very close and no drug can pass. But the thing with LSD is, I mentioned it before, LSD doesn't need to go through these cracks because it's highly lipophilic. So it's actually quite easy for LSD to travel across the membrane of these endothelial cells into the interstitium where the neurons are. So this is true for all the psychedelics. Psilocin would also be able to do it, and so forth. Serotonin, for example, is not lipophilic; it's rather hydrophilic. So serotonin would not be able to do the trick. It would not be able to enter or go across the blood-brain barrier. LSD, however, is able to do it. And once it is here in this interstitium, it is where it belongs, namely in the synaptic cleft, where it induces its pharmacological effects. Yeah, we will speak about the pharmacological effects later, the pharmacodynamic effects, but of course, we also need to ask the question, still speaking about pharmacokinetics, once LSD is within the brain, how is it actually gotten rid of? And this also works via vessels, but this time not via arteries. So arteries bring LSD to the blood, but it's the veins that carry LSD out of the blood. Here you see in blue these veins, and they have all kinds of branches here into the cortex. The smaller branches are called venules. This is where LSD would basically find its way through the membranes into the veins, and then being transported to these bigger veins, which you see up here. This is the so-called superior sagittal sinus, and actually it transverses both hemispheres. It's a very massive vessel. Here, all of the LSD is being carried to, and then it would be transported further across other sinuses, would go down to the internal jugular vein, and then would again find its way to the heart, to the lungs, to the heart, and then via the systematic circulation go to the liver and the kidneys. It's important to know that a lot of LSD molecules actually can travel the body hundreds of times. So it's a matter of time that more and more LSD molecules would be metabolized in the liver and then being gotten rid of by the kidney or by other organs. A minor part, so apart from the removal of the drug by the veins, a minor part can also be might also be removed by the lymphatic system or by the lymphatic system, and certain proteins such as aquaporin 4 might facilitate the exit of drugs by the lymphatic system. But it is assumed that the major pathway of LSD leaving the brain is probably via the sinuses. Okay. When there, of course, this was now basically explained on what we know about pharmacokinetics in general, but of course, people also want to do more specific research on the drug itself, and this was done in the early days by radioactively labeling LSD and then injecting it into the cat or into the rat, and then you just need to visualize the radioactivity, and then you can see at which time point does LSD accumulate in which areas. And what they actually found in the cat, that it would highly accumulate in the bile. This already tells us something about the excretion. We will talk about this in a bit. We will find a lot in the plasma, of course, compartment number 1, we spoke about it. And medium levels in the lungs, the liver, the kidney, and the brain. So these are all areas which are well perfused. LSD always goes with the blood, and less LSD we would find in the intestines, in the spleen, in the heart, and the muscle, and the fat. So this is rather the peripheral compartment where it's more difficult actually for blood to find its way to. Similar things have also been shown in the rat, where radioactively labeled LSD was given to the rat, injected, and it would also highly accumulate in liver, kidney, lungs, less in spleen, heart, and brain. So these are, again, the well-perfused areas, and only little of it in fat or in muscles. So this is actually well in line with what we generally know about the distribution of drugs that, mainly whose destinies mainly are determined by the blood flow, basically. In humans, so these were animal studies. In human studies, what people often do is such a plasma curve, which is very interesting. So you would give LSD to your patients, and then you would basically take the plasma at several time points, and then you can basically see at which. So this is the absorption phase, basically. So how long does it take for LSD to be absorbed? And this would then be the elimination phase. And here you can determine parameters such as the Cmax. This is basically the maximal concentration you find in the blood after application of the drug. You see it's here in this range. You can also determine the time point when the peak is. And this is very often correlated with the peak of the effects, which makes sense. The more drug is in the plasma, the more likely it is that it also finds its way into the brain then. You can also determine the half-life of the drug, and then you can, if the drug follows first-order kinetics, basically predict how much it will take until the drug is cleared from the system. Which first-order kinetics is usually that with every half-life, which is 3.6 hours, 50% of the drug within the body are being gotten rid of. So if you at the beginning have 4 nanograms, after 1 hour you have 2 nanograms, after one half-life you have 2 nanogram, after another half-life you would have 1 nanogram and so forth. So this is what first order kinetics means. So this is something that helps you predict the course of the drug effect. And also when you have drugs where you want to repeatedly apply something, a plasma curve can give you an idea when it's best to reapply the drug, so to speak. We have now spoken about absorption. We have spoken about distribution. The next step we want to talk about is the metabolism. A very important parameter when it comes to the metabolism is actually bioavailability, or F. Basically, the question is, if you consume LSD by mouse, for example, how much of it would actually find its way into the system, or how much is actually destroyed by the liver in the first round? And we know for LSD that the bioavailability is relatively high, so we can actually say that most of the LSD we consume also finds its way into the system, and only little is being metabolized in the first round. So it needs multiple rounds for LSD to be metabolized. In pharmacology, we differentiate phase 1 metabolism and phase 2 metabolism. For LSD, we know there are about, 20 or 30 different metabolites. So the pharmacology of this drug is very complex. So I only chose a few examples for you. Here you got the original LSD molecule. One thing the liver does in phase 1 is basically add a keto and a hydroxy group. This is so via hydroxylation and oxidation. This is meant to increase the hydrophilicity of the drug, so it's more easy to get rid of it in the urine. This is the idea behind it. It's also meant, basically, if this is the active molecule, the liver wants basically to change the molecule by adding these groups, so it's not active anymore. So this is the evolutionary idea behind it. Then there is a phase 2. So once you, for example, this is another metabolite, it's called 13th or 14th hydroxy LSD. So this is one metabolite, this is another one. After this phase 1 has taken place, you would basically add another thing to the drug, which is a glucuronic acid, which is being attached to this hydroxy group, and then you end up with an LSD glucuronide. And this is also meant to make the drug more hydrophilic, so you can get rid of it easily. So these are examples of how the body actually deals with LSD to get rid of it while it travels via distribution, the body again and again and again. And if we have a look at the plasma curve, then you can estimate actually how long it takes for liver, kidney, and bile to get rid of LSD. Something that's very interesting to mention in the context of metabolism as well is psychedelic prodrugs. You probably have heard of them. Psilocybin, for example, psychedelic and magic mushrooms is such a prodrug. As you probably know, psilocybin itself is not able to pass the blood-brain barrier. This is due to the fact that it has a very low logP value. This means it is not really lipophilic. In order to get into the brain, you need to be lipophilic. And psilocybin is then dephosphorylated to psilocin, and this drug has a higher lipophilicity, and that's why psilocin is then actually able to enter the brain and induce the psychedelic effect. Importantly, so this is called a prodrug. Psilocybin is the prodrug for psilocin, basically. Importantly, the fact that psilocybin is a prodrug does not necessarily mean it's not active. It is indeed not able to go into the brain, but it actually binds to a lot of receptors, which also can be found in the peripheral system. And as to this, it's possible that psilocybin in the periphery has effects. This is not very well studied, but chances are that psilocybin is not totally inactive, at least not in the peripheral system. Other examples for prodrugs are these new LSD versions, which you probably have heard of as well, are currently being synthesized to somehow circumvent the law. LSD is illegal, so that's why a group is attached to it. And once this new drug is then made illegal, then another group is attached to it, and this is like a cat and mouse game. And the idea is here that these groups, this 1P group, 1B, and 1V group, and it goes on, is basically cleaved by the liver to liberate LSD. And thereby you would consume it. They are all by now all illegal, but they used to be legal. So the idea is here, I consume a legal drug, and then within my body, the illegal drug is being synthesized, but then you don't have a problem. So this is basically the idea. Then there's actually within the field of tryptamines and phenethylamines, there are really a lot of prodrugs, but another one would be psilocybin. I think it was synthesized by Nichols, I'm not completely sure. And this drug is also thought to be a precursor to psilocin. So speaking about metabolism, prodrugs are important to have in mind. LSD, however, is thought not to be a prodrug, but to be active itself. Okay, how is LSD excreted? This has also been investigated in humans. A lot of the research has been done in Switzerland, actually, and it was found that if we consume LSD orally, only 1% of this LSD would find its way into the urine. And 60% of the LSD consumed would find its way into the urine by this metabolite we've already talked about. This tells us two things. Number one, either LSD is massively metabolized and or LSD and its metabolites might be excreted via another pathway as well. I personally believe it's a little bit in between. In these human studies, they very often only investigate LSD and one or two metabolites, but we know that there are much more. So it's possible that other metabolites can also be found in the urine. But we know from rat research, again with the radioactively labeled LSD, that a lot of the radioligand is actually recovered within the bile and only 5% in the urine. And the bile actually leads the drug into the feces. This is basically what you see here on the right. We got the liver. Here you have basically the bile as it is generated in the liver. This is being collected here in the gallbladder, and then it would be carried down into the intestines. And here it can basically be eliminated by adding it to the feces. And this is very often done with LSD glucuronides, and this has also been reconfirmed in this rat study. So although this kind of research is still lacking in humans, so most of the human research always focuses on the urine. It's not unlikely that a lot of the LSD and or its metabolites can actually be recovered in the feces more than in the urine. But this will probably need more research to have a clearer picture on that. Okay, gut. But what does it do in the brain? This is a question of pharmacodynamics, actually, and if you are interested in this, I will link a video here or there. I'll try to link a video, and you can learn about what it actually does in the brain after it has traveled to the brain. Up to then, take care.